Hello and welcome to our only video of Bio with Geno, where we're making biology fun. My name is Geno, and in this video I am going to teach you more about muscle contraction. Now let us start.
This is a sarcomere, the basic function unit of muscle contraction. A sarcomere is made out of filaments. The thick filament, myosin, and the thin filament, actin. The motor neuron releases neurotransmitter that stimulates calcium release. When that happens, the filaments slide against each other, shortening the sarcomere and contracting the muscles. The following steps are involved in muscle contraction. Step 1. The sequence of events leading to contraction is initiated somewhere in the central nervous system, either as voluntary activity from the brain or as reflex activity from the spinal cord. Step 2. A motor neuron in the ventral horn of the spinal cord is activated, and an action potential passes outward in a ventral root of the spinal cord. Step 3. The action branches to supply a number of muscle fibers called a motor unit, and the action potential is conveyed to a motor end plate on each muscle fiber. Step 4. At the motor end plate, the action potential causes the release of pockets of acetylcholine into the synaptic clefts on the surface of the muscle fiber. Step 5. Acetylcholine causes the electrical resting potential under the motor end plate to change, and this then initiates an action potential which passes in both directions along the surface of the muscle fiber. Step 6. At the opening of each transverse tubule onto the muscle fiber surface, the action potential spreads inside the muscle fiber. Step 7. At each point where a transverse tubule touches part of the sarcoplasmic reticulum, it causes the sarcoplasmic reticulum to release calcium ions. Finally, the calcium ions result in movement of troponin and tropomyosin on their thin filaments, and this enables the myosin molecule head to grab and swivel their way along the thin filament. This is the driving force of muscle contraction.